Hello friends, this video on NEET Reproduction is brought to you by examfear.com. No more fear from exam. Now, how do we categorize embryo sacs? Now, embryo sacs are all the same because all the embryo sacs, what's embryo sac? So, it's a sac-like structure which contains the embryo, which is going to contain the embryo later. So, initially it contains the egg cell and then the male gamete comes and fertilizes the egg cell, and then the fertilized egg cell gradually develops into the embryo. And that is why this sac-like structure is called embryo sac. Now, based on the number of megaspores, embryo sacs can be categorized into three types. So, what are those three types? First type is monosporic, second is bisporic, and third is tetrasporic. Now, monosporic, what do we mean by mono? Mono means 1, bi means 2, and tetra means 4. So does that simply mean that an embryo sac with 1 megaspore is monosporic, 2 megaspore is bisporic, and 4 megaspore is tetrasporic? Does that simply mean that? Let us have a detailed look. So first we will talk about the monosporic embryo sac. So what happens in monosporic embryo sac? Now basically inside the embryo sac, how is megaspore formed? So megaspores are formed from the megaspore mother cell. So megaspore mother cell, which is also abbreviated as MMC, that is megaspore mother cell, and this megaspore mother cell is a diploid cell. So this megaspore mother cell will undergo meiosis or reductional division and lead to the formation of haploid megaspores. So what happened? Meiosis of megaspore mother cell, which is diploid, will give rise to 4 haploid megaspores. So that means here we are having how many megaspores? 4 megaspores. But why is it monosporic? That is because 3 out of these 4 haploid megaspores towards the micropylar end, they undergo programmed cell death. So they basically degenerate. So how many functional megaspore exists? Because 3 out of 4 gone. So how many are left? Only 1. So that means only 1 functional megaspore exists. So basically what is finally existing inside the embryo sac? So finally you have only 1 haploid functional megaspore, and that is why this is called monosporic, because you have 1 functional haploid megaspore. So that is monosporic. Now let us move on to bisporic. So in case of bisporic, what happens? Again, here also meiosis of the diploid megaspore mother cell will take place. So the process will still remain the same, because for the formation of megaspores, meiosis of megaspore mother cell has to happen. So that's the process. So that has to happen. So here also the same thing would happen, but the result in this case is quite different. So like in the earlier case, 4 haploid megaspores were formed, but in this case, 2 megaspores are formed with 2 haploid nuclei each. So like here, it was like 4 haploid megaspores was formed. So 4 haploid megaspores with 1 nuclei each. But in this case, 2 megaspores are formed, but each of them have 2 haploid nuclei, something like this. So here, even though the same process is taking place, but the result is different. So complete division is not happening basically. So inside 1 megaspore, you have 2 haploid nuclei. So that's how the division is taking place. Now in this case, what will happen? What will finally exist? So in this case, again the megaspore near the micropylar end will degenerate. So we will remain with 1 functional megaspore, but this time this 1 functional megaspore has 2 haploid nuclei. So that is why this is bisporic. So basically you see, it is not directly about the number of megaspores that you have, because whether it is monosporic or bisporic, you always have 1 functional megaspore. But in case of monosporic, that 1 functional megaspore has 1 haploid nuclei. But in case of bisporic, that 1 functional megaspore has 2 haploid nuclei. So that is why it is bisporic. Now, a simple question might come to your mind that why is this difference in bisporic? That is, why is the result of meiosis like this in case of bisporic embryo sac? Now, that is because after meiosis, now when we talk about meiosis, meiosis has meiosis 1, meiosis 2. So by end of meiosis 2, the cell number, kind of it becomes half. But in this case, what's happening is there is no cytoplasmic division after meiosis 2, because only when there will be cytoplasmic division, these two nuclei would divide. But that cytoplasmic division is missing in case of the bisporic embryo sacs. In fact, since whenever the cytoplasmic division doesn't happen, there only we get bisporic embryo sac. Now, could you guess something about the tetrasporic embryo sac? So here we are going to have one functional megaspore with 4 haploid nuclei. Yes, absolutely. So in this case, a single megaspore exists with 4 haploid nuclei. So everywhere it is going to be just one single megaspore. It is just that the number of haploid nuclei inside that megaspore can be 1, 2 or 4. So now, why this difference in tetrasporic? So what is happening in tetrasporic? Now in tetrasporic, the cell plates do not form at all after meiosis 1 and 2. And what are cell plates? Cell plates are those walls which actually divides two daughter cells. So those walls themselves are not forming. Since the walls are not forming, so obviously these nuclei, they are not separating out. Right, so everything is remaining inside one single megaspore, and in such case we call this as a tetrasporic embryo sac. So these are the 3 different categories of embryo sacs. So let us do a quick recap of whatever we have learned. So here we have a simple diagram which will help you to understand the difference between the 3. So everywhere you start with a megaspore mother cell which is diploid. So everywhere it is the same megaspore mother cell. After that, what happened? The megaspore mother cell undergoes meiosis to form the megaspore. So meiosis has 2 phases, meiosis 1 and meiosis 2. So in case of monosporic, what happens? At the end of meiosis 1, it divides into 2 halves. 2 halves in the sense, a cell plate divides this one nuclei into 2 nuclei. So if you look at this carefully, that at the end of meiosis 1, the output is same in monosporic as well as bisporic. It seems that everything will happen like this. But by the end of meiosis 2, in case of monosporic, it got divided into 4 haploid nuclei. But in case of bisporic, it just got divided into 2 and each containing 2 haploid nuclei. Right? And how was it different in case of tetrasporic? In tetrasporic, this cell plate, this is known as the cell plate, which divides the daughter cell. So if you see, in case of tetrasporic, cell plates did not form even after meiosis 1. So after meiosis 1 or after meiosis 2, no cell plate has formed. And that is why it is like this. So the end result of tetrasporic is one megaspore with 4 haploid nuclei, bisporic 1 megaspore with 2 haploid nuclei, monosporic 1 megaspore with 1 haploid nuclei, because the rest 3 degenerate. In this case, the other one also degenerate. So this diagram is a simpler one to understand the concept of monosporic, bisporic and tetrasporic embryo sac. And I hope that you understood that why this difference, because in case of bisporic, proper cytoplasmic division doesn't happen after meiosis 2. That is why it is like this. In case of tetrasporic, the cell plates themselves do not form after meiosis 1 and 2, and therefore they never divide into the haploid nuclei separately.
Thank you. Please visit examfear.com for free quality education. You can learn with a simple 4-step learning process wherein you can watch video lessons, you can ask your questions, you can refer notes, and you can take a free online test. We have content for class 6 to 12 on physics, chemistry, mathematics, and biology along with practical videos. So please subscribe to our channel for daily updates. Thank you.